Let's discuss on structure and functions of hemoglobin. Hemoglobin is a conjugated protein where it is a tetrameric allosteric protein due to the it has got 2 alpha and 2 beta globin chain. Hemoglobin is made up of heme and globin component. Heme is an porphyrin ring and globin is polypeptide chain. The red color of blood is due to the heme, which contains porphyrin molecule, protoporphyrin 9. With iron at its center, each gram of hemoglobin contains 3.4 milligrams of iron. Hemoglobin found in red blood cells carries oxygen from lungs to tissues and from tissues to the lungs. Here is the tentative structure of heme. So it is a porphyrin ring. It is in the protoporphyrin 9th variant. So we can see it here. It does consist of 4 pyrrole rings. Each of these pyrroles are connected with each other via methylene bridge. So these are methylene bridges. 1, 2, 3, 4. Then 4 nitrogens, they are interiorly directed. And the pyrrole rings are named as 1, 2, 3, and 4. And bridges, we can call them alpha, beta, gamma, and delta. So this is how it can be named. In the center, iron is fixed through coordination bond, and coordination bonding is via 4 pyrrole nitrogens and 2 other ligands. So pyrrole ring is better numbered 1, 2, 3, and 4. The bridges, methylene bridges, they are named as alpha, beta, gamma, and delta, named here. And these possible sites for substitutions, they are named 1, 2, they are exposed, outwardly exposed sites. So 1, 2, 3, 4, 5, 6, 7, and total 8 sites are available where the possibility of substitution is there. So on the basis of number of substituents from first to eighth positions, there can be different types of porphyrin systems available into the biological system. And almost all of these are, they are component intermediates of biosynthesis of hemoglobin pathway. You will see heme pathway. So first is uroporphyrin 1. It has AP as in at all 8 sites. There is substitution, alternate substitution of all these constituents. A for acetyl, P for propenyl, M for methanol, and V for vinyl. So here in the uroporphyrin, AP means acetyl and propenyl groups are there. Then in coproporphyrin, there is methyl and propionyl group alternately from 1 to 8th positions are there. Then coproporphyrin 3, MP, 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 and PM. Last is there is lack of alteration, and this is PPM. Then protoporphyrin 3, methionine, methyl group, vinyl, methyl, vinyl, methyl propionyl, then again propionyl and methyl. So this is how, on the basis of different substituents, there are different types of porphyrin rings available in the biological system. So we can better see the structure of hemoglobin, the 4 pyrrole nitrogens. The lone pair of electron, they are coordinating with the iron. It has vacant d orbital in which it will be accommodating its electron via coordination bonding. So 4 bonds, 4 coordination bond via the pyrrole nitrogen, and one coordination site is from the globin histidine site. Then another is also from the histidine, proximal histidine F8 of the globin chain. So this histidine nitrogen, that is also participating in coordination. So this is how all 6 sites are engaged in forming the coordination bonds. So these are the various system present in the living beings where the porphyrin ring is available: hemoglobin, myoglobins, cytochromes, peroxidase, catalase, tryptophan, pyrrolase, nitric oxide synthase. Heme can be synthesized by almost all the tissues in the body, and it is synthesized in the normoblast but not in the matured ones. The pathway is partly cytoplasmic and partly mitochondrial. We will see subsequently in the next lecture about the biosynthetic pathway of heme. Myoglobin, it is another pigment synthesized in the muscle cells that gives it a reddish brown color. Temporarily stores oxygen, which reduces the need for continuous supply during the muscle contraction. So instead of four globin chain, there is only one globin chain is there and one porphyrin ring is there. So this is the simple structure of myoglobin. So it can upload only one oxygen. So one oxygen molecule can be carried out by the myoglobin. While for the hemoglobin, there are four polypeptide chain and four porphyrin rings are there in the form of heme, and with each heme there is a loading of one oxygen molecule. So, clinical significance of myoglobin. Severe crush injury causes release of myoglobin from the damaged muscles. So, myoglobin is excreted through urine. That condition is known as myoglobinuria, and urine color becomes the dark red. So, it is indicated in severe crush injury. Then myoglobin will be released from the myocardium during the myocardial infarction and is seen in serum. So that's how it is also one of the important marker for cardiac diseases or cardiac disorders. Then serum myoglobin estimation is useful in early detection of myocardial infarction. So one molecule of myoglobin, as I've already told you, can combine with only one molecule of oxygen. So oxygen carrying capacity is very less, 1/4 as compared to heme. In muscles, the oxygen is taken up by the myoglobin for sake of these two respiration. So myoglobin has higher affinity for oxygen than that of hemoglobin. Though affinity is more, but carrying capacity of hemoglobin is always more, as it has four sites for binding the four oxygen molecule. As compared to myoglobin, only one heme site is available for oxygen bonding. So partial pressure of oxygen in tissues is about 30 mm of mercury when the myoglobin is 90% saturated. At this partial pressure of oxygen, hemoglobin saturation will be only 50%. So this makes the difference that it shows the stronger affinity of myoglobin as compared to hemoglobin. In severe physical exercise, partial pressure of oxygen in muscles lowers to 5 mm of mercury when the myoglobin starts releasing all bound oxygen. So there is no shortage of oxygen at the time of strenuous exercise because myoglobin is available to release the bound oxygen. Myoglobin has higher oxygen affinity, while Bohr effect, cooperative effect, and 2,3 BPG effects are absent, which are very much present and very much manifested by hemoglobin. We will subsequently discuss about it. Then globin. While talking about the hemoglobin, as we have seen, normally it has two alpha chain and two beta chains. But there are four variants of hemo where the alpha chain remains the same. Two alpha chains are there, but it can be either beta or it can be either gamma or it can be delta chains. So if in adult hemoglobin, in 90% of total hemoglobin contains A2 B2 system, means two alpha chain and two beta chain. In HB A2 variant, adult 2 variant, there only less than 5% presence is there. It contains alpha 2 and delta 2 chains. Then fetal hemoglobin contains alpha 2 and gamma 2 chains. It is less than 2% in our system and blood. Then HbA1c, that is less than 5% in normal person. So it has A2, B2 bounded with glucose. So adult hemoglobin is around 90%. That includes HbA1 and HbA1c. The four globin polypeptide chains with four hemes are held together and are stabilized by hydrogen bonds. So we can see here there is a prominent role of hydrogen bonding, salt bridges, and van der Waals forces in order to maintain the tertiary and quaternary structure of this hemoglobin. So these are the salt bridges, the four chains held with heme nucleus. They are all attached with each other via the salt bridges. Then protective hydrophobic pockets formed by globin chains. This is the central open cavity, and these are the hydrogen bond in between. So they are an important role played by all these three forces. There is little contact between the 2 alpha and 2 beta chain, but there are many contact points between the alpha and beta chains of dissimilar chain pairs A1, B1 and A2, B2. So we can see it here. What happens? It shows about the cooperative bonding. So once there is a bonding of one oxygen, that leads to the conformational change. Actually, this particular state is known as trans dot tout state. The molecular system, the heme molecule system is very strained. So that condition is known as tout structure. That is the deoxyhemoglobin at the time when there is no oxygen. So once one oxygen binds with any of the heme of the globin, so it leads to conformational change of this whole system, and it make it into the relaxed form. So in this relaxed form, it can easily accept all the remaining three oxygen. This is how the loading of four oxygen happens and leading to the formation of oxyhemoglobin structure. That is all in the uh, relaxed structure form. And in this state, there are broken, we can see the salt bridges are broken, making it relaxed. There is no strain in the system. The cooperative binding of oxygen to hemoglobin. So once I have already told you, once the one oxygen binds to any of the heme of the all these four globin chain, it leads to a conformational change which converts it from the taut form to relaxed form, and that makes the facile addition or binding or the binding of all remaining three oxygen molecules. So, ekdam se, like you can see in the structure, initially there is a little delay in saturation with oxygen, but once the one oxygen is added, immediately there is an no straight raising of the curve and leading to the complete saturation of hemoglobin by binding with all the three remaining three oxygen molecules. So this is how the partial pressure curve of oxygen binding of hemoglobin is sigmoidal. The curve is sigmoidal. While that of myoglobin, it is hyperbolic curve. So the shape of oxygen bonding curve of hemoglobin is sigmoidal because the binding oxygen bonding is cooperative. It shows the cooperativity of the one oxygen molecule. So that facilitates the binding of other three oxygen molecules. So because of cooperativity between the oxygen binding sites, hemoglobin delivers more oxygen to the tissues than would be a non-cooperative protein, that is myoglobin. So in case of myoglobin, there is no as such scenario is seen of cooperativity. Only one oxygen molecule has to be added, and with high affinity, it is attached with the myoglobin. So there is a little delay in release of oxygen right from myoglobin. So it remains in the binded form and the stored form in the tissue sites. So once the oxygen pressure becomes low, lesser than 5 mmHg, then it starts breaking out and releasing the oxygen. So Bohr effect, what is the Bohr effect, which is not being shown by the myoglobin? Only hemoglobin shows this effect. Hemoglobin also transport a significant amount, about 20% of the total H plus ions and carbon dioxide from the tissues to the lungs and the kidneys. So bonding of H plus ion and carbon dioxide to hemoglobin decreases the affinity for oxygen because the affinity of binding of H plus ion and CO2 with Hb is more as compared to oxygen. So once they are binded, so they decrease the affinity of oxygen binding. So that's at relatively low pH and high CO2. We can understand. Again, I'm reading. Low pH means high, H plus ion concentration is high, highly acidic pH, and high concentration of CO2, it in the peripheral sites, the affinity of Hb, hemoglobin for oxygen, is decreased, and this effect is known as Bohr effect. Conversely, what happens in the lungs? In the lungs, there is a reverse scenario. In the lungs, the carbon dioxide is excreted and the blood pH consequently rises. That means from acidity to alkalinity, the H plus ion concentration decreases in the lungs. So the affinity of hemoglobin for oxygen increase in turn because pH lowers, carbon dioxide is removed. So that leads to higher affinity of oxygen for hemoglobin. So inverse relation between the carbon dioxide H plus affinity and the oxygen affinity. So this effect of pH and carbon dioxide concentration on the binding and release of oxygen by hemoglobin is known as Bohr effect. So in this diagram also we can see it here. What is happening in the peripheral tissues? This is the oxygenated hemoglobin. So it is dividing up into, because oxygen pressure is low in the peripheral side, so oxygen is downloaded. Hemoglobin gets freed immediately. It is being caught by H plus ions, which are being released because carbon dioxide is in higher concentration because of metabolic activities. Cellular respiration is happening. So continuous production of carbon dioxide. Carbon dioxide binds with water. In the presence of high concentration of carbonic anhydrase, it converts up into H2CO3, which again broken up into H plus ion and bicarbonate ions. So these H plus ions are now being added to hemoglobin, forming the hemoglobinic acid, right? So this is the hemoglobin blended with H plus ion, and in this form, it is being carried right from peripheral side to the lungs via blood. And the same way, the bicarbonate ion concentration is also increased in the peripheral tissue. So right from peripheral tissue to the blood, and of course to the lungs also. So what is happening here? See this in the lungs, this hemoglobinic acid is broken up into H plus ion. Because the oxygen concentration is higher, now the H plus ion has to be detached from the hemoglobin, and in place oxygen is now being added because high pressure, high concentration of oxygen is there. So this hemoglobin now again converted up into the oxygenated hemoglobin. H plus ions are released, and these H plus ion again combined with bicarbonate ion, leading to the formation of carbonic acid. Again, in the presence of carbonic anhydrase enzyme, it's broken up into the carbon dioxide and water. This carbon dioxide is now exhaled out. So this is how the parallel transportation of oxygen right from lungs to tissues and from tissues to lungs, the transportation of H plus ion and carbon dioxide takes place. So chloride shift, this is the scenario of chloride shift in the erythrocyte because of the higher concentration of carbonic anhydrase enzyme. So this they combined with the H2O combined with carbon dioxide lead to the formation of carbonic acid. So there is a huge production of bicarbonate ions, NH plus ion, and these H plus ion wind up with hemoglobin. They form the hemoglobinic acid, and this bicarbonate is in high concentration, so it starts coming out of the erythrocyte. So right from erythrocyte to the plasma, and in turn, in exchange of this bicarbonate ion, the chlorides gets inside of the RBCs in order to maintain the ion balance. So this is known as uh, chloride shift. This chloride comes from where? Because a lot of NaCl is present in the plasma. This is the plasma tissue in the plasma. So this chloride is in the form of NaCl. So once the bicarbonate reach there, sodium binds with bicarbonate. It forms the sodium bicarbonate. Chloride is released, and this chloride is all set to move to the erythrocyte in order to maintain the ion balance, because a lot of bicarbonate is now shifted out, and in turn the chloride has to shift inside of the erythrocyte. This phenomena is also known as Hamburg phenomena. So in all hypoxic state, the oxygen affinity is decreased with a shift in oxygen dissociation curve to the right and an increase in the 2,3 BPG inside the RBCs. While discussing the glycolysis, I had a comprehensive discussion onto this bis phosphoglycerate, 2,3 bis phosphoglycerate in the shunt pathway. Then adaptation to the high altitude where the partial pressure of oxygen is low. It includes the increased pulmonary ventilation, polycythemia, and increase in 2,3 BPG level with a shift in oxygen dissociation curve to the right. So those both your a higher concentration of this 2,3 BPG and at high altitude where the PO2 is low. So oxygen dissociation curves always turn or moves to right. So in anemia where the total concentration of hemoglobin is reduced, increased oxygen unloading alone will ensure the proper oxygenation of tissues. So in many cases, 2,3 BPG level varies inversely as the hemoglobin concentration increases. So in chronic pulmonary diseases, in cyanotic cardiac diseases, and also the 2,3 BPG level is increased, ensuring the maximum unloading of oxygen to the tissue. So it has got a vast and very important role in downloading the oxygen to the site where oxygen is very much required. So the red cells 2,3 BPG in the red cell, the 2,3 BPG level is decreased in acidosis and increases in alkalosis. Hence, the expected shift in oxygen dissociation curve to the right or left is not observed. Transfusion of large volume of stored blood, which has a low level of 2,3 BPG, can lead to sudden hypoxia, since it can cause a left shift of the oxygen dissociation curve.